Well, thanks for inviting me, Mark, and thanks for everyone for showing up. I should say I've only recently started in Leeds, and it is a grown-up job, so I am looking for PhD students and postdocs, etc. So I have no money right now, but if you're interested, come and chat to me afterwards. And whether or not this talk is interesting to you might be a reason to decide one way or the other. So I'm going to start by acknowledging my invisible co-authors, effectively. So Katie Davis helped me build the big trees you're going to see. Dave Babst and Matt Friedman helped me timescale them. Dave also helped with the birth-death model, as you're going to see in the last section of the talk. And then Laura is sort of my collaborator for all the paleobiography stuff. And so most of this work comes out of my previous job, which was in Australia. So that was funded by Macquarie University and the Australian Research Council. And Laura and I also got grants from Nescent, so we spent a month in a windowless room in North Carolina writing code. But it was quite useful. So in order to do a seminar, what I've basically done is stuck together 3 conference talks. So unfortunately, some of you will probably have seen at least one of them before. So the themes will be very obvious, but hopefully also the links will. So I'm going to talk first of all about how I build big trees, then about what we can use them for in terms of paleobiogeography, and then what we can use them for in terms of palaeobiodiversity. So I'm going to start by talking about how we build big trees. And a good place to start this talk in general is the Sol figure from Darwin's Origin of Species, which was, of course, effectively a phylogenetic tree. And more generally, of course, this was kind of how Darwin pictured evolution. And where evolution really happens is effectively along the branches of some phylogenetic tree. We don't necessarily know we've got the right one, of course, but that's kind of a framework we should be using to sort of ask and answer macroevolutionary questions. So the kinds of questions we can ask with big trees, these are just some examples. There are lots. So you might look at the sizes of different clades in terms of the number of species in them, and we can ask questions why are sort of sister clades relatively disparate in terms of the number of species they have and whether or not there are diversification shifts on the tree. We can ask things about traits, so we could look at something like body size, and we can fit models once we've got tip data in a tree to look at whether or not there's trends, for example, in body size. We can use it to look at paleobiogeography or biogeography in general. So effectively dispersal events and extirpations and extinctions, etc., are all going to happen on the phylogenetic tree. And then, of course, as paleontologists, we're often looking at extinct groups, so we can look at how extinction events might prune the tree of life. So an example at the top here would be sort of snipping away at the tips of the tree, whereas the other example at the bottom there, you're sort of hacking away a whole sort of deeper branch and getting rid of a whole clade. But if we want to look at a lot of paleontological groups, they can be very large in terms of the number of species that we might want to look at in terms of tips. So for example, an estimate for Mesozoic dinosaurs might be something like 1,200 species. We can go bigger if we look at graptolites. There's about 2,000 species. If we look at just extinct mammals, there's probably something like 5,000 species. There's certainly more extinct genera of mammals than there are living genera, regardless of what that means. Then we could think about something like trilobites. I'd love to play with a trilobite tree, but that's like 20,000 species. We're probably a very long way away from doing that. So ideally then, for each of these groups, if we wanted to look at their macroevolution, we'd have a tree with all of those species as tips. But that can be quite hard for us as paleontologists, because we really only have morphology to look at, and it's very hard to build up a morphological data set in the same way you might do with molecular data and something like GenBank. So this is an example of some of the biggest trees we have so far. It's probably a bit out of date, but it sort of makes the right point. So these are sort of 3 very large matrices. The numbers in red are the number of taxa. In blue is the number of characters. So this is Gautier et al.'s sort of big squamate tree, Livesey and Zusi's big bird tree, and then the O'Leary et al. Mammal tree. And these are all huge efforts. They took a lot of work, a lot of labor to produce them. But you see the numbers in red are sort of order of magnitude lower than the kinds of things we might want to look at some of those groups I just mentioned. So if we're going to build big trees, there's really only one practical alternative at the moment, and that's to build some kind of composite topology by taking a bunch of smaller trees from the matrices we do have and combining them together into some larger tree. So I would say there's probably 3 main ways of doing that right now. So and I'm going to sort of express these as kind of pipelines. So the first one is an informal supertree. So you take published topologies, you digitize them in some form, Then there's some kind of often opaque sort of cutting and pasting of those trees together, and then you get something like a consensus tree out at the end. An alternative to that is a formal supertree. So normally then you collect more trees than you would for an informal supertree. You use an algorithm to kind of stick them together, and then you normally get a set of topologies out at the end, which allows you to quantify phylogenetic uncertainty. So you can probably tell I already favor this one over the top one. But the kind of way I want to build trees now, and this is sort of something I've instituted relatively recently, is what I'm calling a metatree pipeline. So the big difference is that instead of starting with a published tree, which is a figure in a paper, you go and find the original matrix, and then you reanalyze that. And that gives you sort of basically more information to take forwards, but there's lots of other sort of advantages. So you could decide, for example, instead of use parsimony, you might want to use a Bayesian approach. We still sort of stitch it together with an algorithm. We still get basically a range of trees out at the end that allows us to quantify uncertainty. So I'm obviously biased, and I think this is the best way to do it. But why is that? Well, one of them is there's some problems we can get with entering trees. So here's a very simple one. So this is the published tree. If you go to the actual figure in the paper from Xu et al. 2013, and here is what you get if you reanalyze the matrix on the right. And the difference is basically a lot of times in the published tree, people have pruned out the outgroup taxa. So these we can retain them if we've gone through the Metatree pipeline. And this is important information because those outgroups tell you about overlap, and that really helps you stitch together your topologies later on. So that's one advantage from doing a metatree over a supertree. Another one, this is probably not the nicest figure in the world, but what I'm doing here is comparing the number of possible bifurcating resolutions of a strict consensus tree with the number of trees that are actually used to produce it. And the point here is basically that they all fall below this one-to-one line. So if you're doing an informal supertree, you get some kind of consensus. You might, in order to do analysis, need bifurcating trees, so you'd randomly resolve those. But if you do that, what you can often find is you get a set of trees that would be considered suboptimal based on the original analyses that we used to produce them. And in the same vein, if you're entering a tree from a paper, it's off a consensus tree, so you're kind of losing this information, and you might infer clades are optimal that are not optimal. So more generally, basically, there's a one-way flow of information going from your set of trees that you use to produce your consensus. You can't go back from your consensus to get your original trees. So Metatree pipeline allows you to retain your original set of optimal topologies. So my Metatree pipeline looks something like this. Don't worry about reading everything right now. I'm going to go through all of these boxes. And I'm going to use dinosaurs as the example to explain them, because that's basically the tree I'm going to use in the next two parts of the talk. So the first thing I do is go out and collect data. So I just go into the literature. I find basically phylogenies. I retain the ones that have produced some kind of cladistic matrix. And I put these all on my website once they're up, so you can use them if you want. So currently, this is slightly out of date. There's almost 2,500 data sets on there. There's more references listed, but it takes me a while to sort of process the data. But if you go on, you'll find lots of basically individual references that look something like this. So there's the original kind of reference in sort of Harvard format. And then there's a bunch of links if I've got the data set underneath. So these basically correspond to the actual matrix in Nexus and TNT format. At the moment, I've only analyzed them using parsimony, so there's the most parsimonious trees, strict consensus tree. There's an XML file that has lots of metadata, which is important. But the main thing in terms of building composite phylogenies is there's an MRP file. So MRP stands for matrix representation with parsimony. And what that does is it's a way of sort of encoding a tree as characters that you can then sort of combine all your trees together in one giant matrix to sort of infer your topology at the end. So how this works in practice is if we wanted to encode this node in here, which is the clade BC, we would have a character in our matrix. The taxa that are present in the clade, we'd code 1. Everything else, we'd code 0. We can do the same for the DE clade and for the BCDE clade. We don't need to do it for the roots because everything would basically be coded 1, so it's an invariant character. It doesn't hold any information. But the idea here simply is if you give this matrix to something like PowerPod TNT, it will return your original tree, so you're retaining the information in a sort of one-to-one way. So with my Metatree pipeline, I've come up against two big challenges with this. So one is that if I want all of the MPTs, sometimes there's a memory limit in your tree search. You can't get all of them. And the other is if you turn all of your trees into MRP files, you end up with so many characters, you have this unwieldy sort of massive data file at the end. Now, I've got elegant solutions to both of these problems, or at least what I think is elegant solutions, but I'm not going to tell you because it's probably far too boring. But if you're interested, you can talk to me later. So the next step is probably the most important one. It's certainly what takes the most time, and it's called taxonomic reconciliation. So what you're doing there is taking the names that are on your source trees and validating them against some kind of single taxonomy that you're going to use. So in practice, what that means is you're taking outgroups and removing them. So if you're interested in dinosaurs, you don't want any crocodiles in there, for example. You have to deal with things like shortened names. So T-Rex should be Tyrannosaurus Rex. You have to deal with synonymies. So taxonomy is constantly changing. Some taxa become invalid and replaced by a senior synonym. If you get a valid taxon, this is the nicest case. You don't have to do anything with it. Invalid taxa, things like nomen dubiums, you end up having to remove. Then there's a sort of special case of higher taxa where you want to replace them with the constituent species. So this is by far where most of the effort goes, because it has to be done with your eyes and a brain. You can't really make a computer do this, because a computer won't understand that T-Rex is meant to be Tyrannosaurus Rex, for example. Or if someone has a typo, things like that. You need to kind of do that manually. So here's an example of why it matters. This is an example of it going horribly wrong. So this is from a published paper. I'm not going to share what that paper is. But if you know your taxonomy, you'll notice straight away there's some problems in here. So Sphenodon is a rhynchocephalian. It's a sister group to squamates. Gophyrosaurus there is a lizard, which is a squamate. Then we've got squamata as a taxon itself. And then weirdly, these are all collected together in a single clade of squamata. So none of that makes sense. Then down here, we've got another OTU called Lepidosauромorpha, which is the clade that unites rhynchocephalians and squamates. And we see it's nowhere near the rest of those taxa in this tree. So this is a big problem. That means this tree, you can't really use it for anything. The way to kind of avoid this really is to do everything at the species level. That means you're less likely to end up with these problems because to a computer, squamata and Gophyrosaurus, again, it thinks they're separate OTUs, but if you're a human being and you understand taxonomy, you know those two things shouldn't be separate OTUs in your data set. So what I've also done is basically the one true taxonomy that I'm using is essentially the Palaeobiology database. And there's a couple of advantages to doing this. So this is effectively a meta-taxonomy, but it's also dynamic, so that the Palaeobiology database is continually updated, which means that your taxa can be continually updated as well. So this allows you to resolve sort of synonymies to some degree automatically, but also it means you'll link through to occurrence data, so stuff on time and space for your tips that obviously make downstream analyses much easier. So just to give you an idea, I said this is the hardest part in terms of labor. So for the dinosaur tree, you're going to see there's 961 valid species. But I had to go through 13,000 individually different names in order to get down to that list. So the next thing we do is we want to filter out some of our data. So for example, we don't want supertrees to be in our metatree. We don't want things that are designed to look at trace fossils to be in there, unless that's what we're interested in. We don't want some data sets that are designed to be acladistic. They're using characters, but they're not trying to infer phylogeny from them, so we probably shouldn't be taking them forwards. But another big question and a criticism of supertree methods is that they can use very old data sets alongside new data sets, and maybe you don't want to do that. So what I have at the moment is a protocol that uses taxonomic coverage as a way of deciding where we should sort of cut off what we're trying to do. So imagine the y-axis on here is basically the total number of species you can include, and then the x-axis is year of publication, and it's from that year to the present. So let's say this was 2016 on this bar here. If we only took the trees from 2016, we wouldn't get all the species we want. Even if we went back to 2015, we'd get more, but we wouldn't get everything. We'd have to go back to this point here, where we've got all the species we can possibly get. And then if we go further back in time, we're not adding any extra taxa. And so for the dinosaurs, I think it came out somewhere like 1998. And what that effectively means is there was some valid taxon included in a tree back then that's never been in a phylogenetic analysis since. So the next thing I do is add what I call a taxonotree. So taxonomy can basically be turned into a tree, and so it would normally be poorly resolved. It looks something like this. So you have your species in blue, then you have your nodes represent higher or supra-specific taxa that they can be assigned to. So the reason to do this is two things. One, this is what I use when I'm doing my reconciliation to decide what the constituent species are of a higher taxon. So if a family is an OTU, I replace it with the species that belong to it. But the other thing is I also put this in as an additional source tree. So the reason to do that is it means sometimes we don't have any information about overlap, but for example, we're all quite happy with how ornithischians, sauropodomorphs, and theropods are related to each other, so I think it's fine to probably put in a taxonomy sort of in there. But I don't have it constrain the analysis, which is what a lot of supertree people do, and I have it down-weighted. So taxonomy only comes into play when we either have no information or the information we do have is conflicting. So the next thing I do is weight the data. And the most obvious sort of problem with morphological matrices is that they're constantly reused, so they're not really independent data sets. So we kind of realized this back in 2008 when we were doing the previous version of the Dinosaur supertree. And so we dealt with this by saying, well, if a matrix is reused, it makes the old one redundant, but sometimes a matrix is used multiple times, and so data sets can have equal claim to independence, and so what we want to do is weight them so that they add up to a single data set. So the other thing that I've done for the Metatree as well, though, is up-weighted recent data sets over older data sets, and that's another way to try and get at that issue of should we be including older things equally with newer things. And I would argue we would hope that science is improving and our sort of estimates of phylogeny are improving, so it's probably a safe thing to do to weight recent things over older things. So the next thing I do is relatively arcane. You might not have heard of it. It's called safe taxonomic reduction. So what this is a way of, prior to inference, you can remove some taxa from your matrix that you know where they're going to come out at the end, because the information they have sort of allows you to know that. And it means when you're inferring your tree, you're sort of reducing the amount of tree space you have to search, and it can speed up your analysis. And in supertree, sort of metatree kind of MRP, matrix representation with parsimony, you find that this happens a lot. And so I'm going to use, or for this I used my clades package, which has a function that does this. And it turned out I could remove 50 OTUs, which is basically 5% of the data with this. So these are reinserted later because we still want them in the tree. We still want as many taxa as we can for whatever macroevolution we do later. But this has sort of helped reduce sort of the tree space size we're searching. So once you've done all of that, you end up with basically a giant matrix of zeros and ones and question marks. And then you can give that to PowerPoint, and you can set it running. In this case, it sort of ran on a workstation for 3 weeks. And eventually, you'll get a big tree at the end. So this is the strict consensus for dinosaurs. Obviously, it's too small names to read, and I'm not going to go through all of the topology. So there are other metatrees that are kind of produced or using similar sort of protocols to this, and there's various other ones that I'm still working on. So the previous Dinosaur supertree from 2008 was using some of these ideas, and it's sort of what I've done here builds on that. I have a crocodile, or a Eusuchian tree, and then the sort of teleost, actinopterygian rather, phylogeny of John Clark, which was in PNAS last year, was also based on a sort of metatree type protocol. So sort of conclusions for this section of the talk. Basically, we need big trees to ask sort of macroevolutionary questions. Morphology is not going to get it done. I would argue the best thing we have right now is the Metatree pipeline, but obviously I'm biased. And it's sort of already basically yielding trees that we can use for macroevolution. So that's kind of one use then I'm going to talk about next is palaeobiogeography. And the prompt for this is essentially what use are dinosaurs? So dinosaurs basically get a lot of press because they're dinosaurs, but if we were trying to be more objective about it, what are dinosaurs really good at that maybe other clades aren't good at? And I would argue perhaps palaeobiogeography is one of the best things they might be good for. And there's a few reasons for that. So first of all is they are cursorial. Obviously, there's a big exception to that we'll get to later. But this means, generally speaking, they can't sort of cross oceans, and so we might expect their fossil record to have some signal of biogeography in terms of barriers to dispersal. Their fossil record also spans a really interesting time in terms of paleogeography, because they start out when Pangaea was intact, and they basically sort of track right through the break of a Pangaea to something like the modern configuration. And as I've just shown, we have a really good handle on their phylogeny, which is important for the kinds of macroevolutionary tests we might want to do. So there's an awful lot of paleobiogeographic approaches out there. This is one from the BioGeoBears package of Nick Matsky. And this is sort of stochastic character mapping of different sort of dispersals and area. The different colors are different areas, so you're seeing different dispersal events across the tree. But this is sort of part of a much larger group of approaches that basically require you to, first of all, take your data and assign everything to a discrete area. And generally, if you're taking what is really continuous data and discretizing it, you're losing some kind of information. So what I wanted to do is try and develop a new approach that used the continuous data of sort of palaeocoordinates to see what kinds of questions we can ask in terms of paleobiogeography. So a place to start for this is just thinking about basic patterns in terms of things like endemism bicariance. And so this is sort of a scenario at the top where you might have the gray box representing continents that were once joined together. They split apart, and the clades, the clade you're looking at ends up with sort of subclades isolated on each continent. So the idea is that if that's the scenario we have, we ought to see geographic distance and phylogenetic distance positively related to each other. So your close relatives are also geographically proximal. In contrast to that, if you have something like an intact Pangaea, if you're dispersing in what I'm going to call a passive way, so think Brownian motion, so just randomly going wherever you want, we'd expect it to be a much more scrambled signal. And so if we correlated phylogenetic distance and geographic distance, we would find no correlation signal. And then finally, of course, we could get a negative correlation. So what might that mean? What that would have to mean in practice is that close relatives are trying to live far apart from each other. So processes such as competitive exclusion might be something that drives that. And so I call that here active dispersal, because it's no longer random where things are dispersing. So in terms of dinosaurs, then, I had three major hypotheses I wanted to test. So first one is that when dinosaurs first appear, they should have this sort of Pangaean pattern of no correlation. And then over the course of the Mesozoic, they should shift to this sort of positive relationship with the clades sort of becoming isolated on continents. But as I suggested earlier, of course, not all dinosaurs are cursorial. Along the way, a clade evolved flight. And so my argument then is maybe that made, for birds, the world more like Pangaea again. The barriers basically didn't exist for birds, or that's the hypothesis, and so they ought to move more towards this no correlation type pattern. So the data I'm using is the tree you just saw, and again, the sort of time and space data, the palaeocoordinates, come from the paleobiology database. So this is where I'm going to plot the results, but I'll just sort of guide you through everything on here first before I bring them up. So on the x-axis, we basically got time from the Triassic on the left through to the end of the Cretaceous on the right. The y-axis is a correlation coefficient, which is just a correlation coefficient. And then on the right here, I've got those three different sort of scenarios that I'm expecting based on those y-axis. So at 0, we ought to have this passive dispersal. The positive is our endemism bicariance, and then the sort of a negative space at the bottom for active dispersal. And at the top there are basically a series of paleogeographic reconstructions to kind of give you a general idea of what's going on. So what we're looking for in terms of my hypothesis then is basically a sort of a ramping up from left to right. And so that is broadly what we see. There's some fluctuations in there, but we see in the early part of the history, our uncertainty envelops the 0 value, and then it moves more into the sort of positive region as we go further to the right. So generally, the size of these error bars relate to sample size, so that's why it gets narrower, because the fossil record gets better, or sampling gets better as we move towards the late Cretaceous. But it does seem to fit generally this passive dispersal moving into the endemism bicariance area. So the next thing is to look at birds, is to split out the non-avian dinosaurs plotted in red here, and then I'm going to bring up the birds shortly. But to look at non-avian dinosaurs on their own, first of all, it's basically exactly the same signal, because for most of their history, they were the only kinds of dinosaur, and then for the rest of the Cretaceous, they certainly dominate in terms of the species that we can sample. So the first bird is still probably unequivocal bird, is still Archaeopteryx in the late Jurassic. But that means basically we have this period here where there's just not big enough sample sizes to say anything at all. And then by the time we have sample sizes, we can do something. We find actually, instead of plotting out closer to this 0 line, they're actually more up in this sort of positive space. So that could be two different things. It could be a bias in that a lot of those birds are going to come from China. So maybe we're just seeing a strange signal. But it could also be biological in the sense of maybe once you evolve flight doesn't mean you can immediately cross an ocean. So perhaps that kind of makes sense biologically that they have a sort of higher level of endemism bicariance because they would have been small things. Generally, large things can disperse further. So then we go through another window in the mid-Cretaceous where we just don't have a big enough sample size to say anything. But it gets most interesting, I think, in the late Cretaceous. So now we find birds do plot below non-avian dinosaurs, but more interestingly, they seem to be moving more into this negative space of active dispersal. Now, the error bars here generally are quite big for birds. Again, the sample sizes aren't very large, so we could probably still say maybe this is passive dispersal. But this could be interesting because it could suggest competitive exclusion is going on, which would be quite cool because these are basically going to be stem birds. That's not the crown, and it's a pattern we might see in crown birds, but maybe it occurred earlier than the living radiation. So what I've essentially done so far is I've kind of told you this pattern corresponds to this process, but we don't actually know that's true because we haven't seen the process happen. So what happens if we kind of invert these arrows and try and simulate the process? Do we see these patterns that I've argued are sort of telling us something about biogeography? So in order to do that, we had to basically, Laura and I built a model from scratch that simulated basically a globe with continents on it. The continents can move around, and then you've got organisms that can move around on the continents. So for someone that's not like super mathematical, this was kind of hard for me. The things that I knew would be tough going in is you have singularities at the pole. So if you've got a dinosaur dispersing east from the North Pole, where is it going? That's kind of a hard one. You've also got what's called a periodic boundary condition, basically where the date line is. You go too far east, suddenly you're in the far west. But those are relatively easy things to deal with mathematically. The things that I wasn't expecting to be hard are things like this. So if I was asking you as a human being, is the red circle on South America, you'd very easily be able to say yes. But if I gave you the coordinates of the red dot and the coordinates that describe the shapefile of South America and told you to write an algorithm that would tell me whether or not that red dot was on South America, you'd probably find it a lot harder, and that's what a computer has to do. So this is known as the point on a polygon problem. And the reason why we needed to solve it is if we're moving our species around on a continent, we have to know if the move is valid or it's ending up in the sea when it's a terrestrial organism. So the other big problem, and this is every video game designer encounters this, is called collision detection. So if we've got continents moving around, we have to have them behave sensibly when they collide. We don't want them passing straight through each other. And here's an example of how it can go wrong. And this is a much harder problem. This one I think I could probably solve now, but this is very tricky. So the way we did solve it is to make it much, much easier. And the way to do that is to use circles for your continents. So now if we want to ask a question, is our point on a polygon, all we need to do is find the great circle distance between the center of our circle and our point. And if that's greater than the radius, it's come off the polygon. If it's less than the radius, it's still on the polygon. And the same kind of thing we can do with collision detection. So we just look at the distance between the two centers of our circles. And if that's less than 2r, then they've collided. But we also wanted to allow them to have some degree of overlap. So we include this parameter we're calling squishiness, which is basically how much they're allowed to overlap before we stop them basically overlapping further. So once we'd sort of put this model together, we basically had a situation where you can pick a bunch of input parameters before you start your simulation. So broadly, these are grouped into what we can input for the continents and what we can input for the organisms. So for the continents, we can pick how many continents we want to have. We can pick how fast we want them to move. We can pick how big they are. We can pick their starting configuration, so we could have them be in a sort of supercontinent like this, or we could have them spread apart, or just some kind of random thing. Then we have another parameter called stickiness that decides how long two continents that are joined stay together or split apart. And then we have that squishiness parameter that sort of allows us to decide how much overlap we want. And some degree of overlap is important because that's what allows things to disperse between continents relatively easily. If we didn't have that, it'd be very hard to disperse between continents when they're in contact. So then we also have parameters for the organisms. So underneath, at the moment, we just have a simple constant rate birth-death model. So the number of new species, or the rate of new species appearing, is the same right through our simulation, same for the rate of extinctions. Then we have what's called a dispersal kernel. So it decides how far an organism can move in a time step, but also what direction it can move. So we can have it be basically any direction is equally likely, or we can force it to be more likely to move, say, east-west, which might be more realistic. We also allow in there what we call sweepstakes dispersal, so there is a small chance an organism can cross an ocean. We know this happens in biology. For example, that's how monkeys got to the New World. But in the simulations we're going to show you, we keep this turned off. So we spent 4 weeks writing the code for this. It's on GitHub if you're really interested. And we gave the package the hilarious name of disperse with a capital R because it's all in R code. So in order to sort of visually check what it's doing, I'm going to show you this hideously colored video. So this is all mathematically happening on a sphere, but this is a projection into a two-dimensional space, so you can see both sides of the planet at once. So effectively, we've got longitude on the x-axis, latitude on the y-axis. So here we have a sort of Pangea configuration at the beginning, and it's sort of, at the moment, it's across the North Pole, but the model is simple, so that doesn't mean anything. There's no ice caps or anything in there. And then that red dot is basically where our clade starts out as a single species. So then we can basically run the simulation. What we see is our clade is diversifying and dispersing, and it's all just with Brownian motion effectively. It's just randomly going where it wants. And we see Pangea is starting to break apart. And it looks basically like continents have measles. So you see the sort of distortion you're getting. These are all circles, but right at the pole, they sort of have these weird elliptical shapes because of the projection, and they look more like circles when they get closer to the equator. This continent is going to end up very lonely soon because that is going to die out, but it's useful because it shows us, there we go, so this is now empty, so no new species can appear in here, but it's going to collide soon and we'll see a biotic interchange, so that's good for us because it shows the model is working. So there we go, more species on it again. So in terms of running those simulations then, there were two scenarios we wanted to look at. So in both scenarios, we start out with a Pangea, a random sort of clade starting point, and then we run the endemism bicariance scenario, so we make stickiness really low so the continent breaks apart, and then the passive dispersal scenario where we make stickiness very high so Pangea stays together. And the idea then is we should find the positive correlation for the endemism bicariance and the no correlation for the passive dispersal. So we didn't simulate active dispersal because it's far more complex and I can't think of a good way to do it. If you have a good way to do it, then tell me. It would be useful to know. And then effectively, these are the results we get. So passive dispersal, we get exactly what we wanted to see. It's a really tight correlation around 0. So the no correlation that we were expecting. So this sort of means we can be reasonably confident that's kind of what goes on when this is the process underneath. And for endemism bicariance, it's definitely skewed into the positive, but we see, unfortunately, the tail does come down and overlaps with our sort of passive dispersal. So what this means in practice is if we find a positive correlation, we can be like very confident that it is endemism bicariance, at least under the assumptions of this model. Whereas if we find no correlation, it's probably passive dispersal, but we can't actually rule out endemism bicariance. So obviously the model is extremely simple. There are lots of ways we could extend it. So one, for example, would be to turn our continent into two circles. So one represents land, one represents shelf. And that means we could look at marine organisms. We could add some ice caps in there. We could add sea level in there, and then this would interact with these other things. So for example, a shelf area could get larger and smaller, ice caps can recede and grow. But we also put some kind of climate in there, at least in a primitive way, so things like Hadley cells, for example, and that might affect how things can disperse. It'd be useful to have some kind of varying temporospatial windows that represent our sampling of the fossil record. At the moment, we're assuming we can see the whole thing, but obviously that's not realistic. We could put some topography in there, so an additional sort of constraint on dispersal. And then our simple constant rate burst death model, we could sort of maybe make a bit more interesting in terms of having, say, an overall carrying capacity, so making it diversity dependent. But we might also want to put in a species area effect in there. So this is all very kind of, it was really hard to write the code we've written, and every one of these it would be super hard to do again, pretty much. So I haven't got to any of them yet, but those are the kinds of things I want to do in future. So we return to our hypotheses. It looks like for an intact Pangea, we are seeing what I expected to see. There is that slight caveat with the simulations that maybe there is actually endemism bicurrence, and we're just in that sort of scenario where it happens to end up looking like no correlation. But towards the end of the Cretaceous, where we are finding that endemism bicurrence signal, the simulations seem to suggest we can be reasonably confident in that. I was completely wrong about birds, but in an interesting way. So early birds being perhaps more endemic than non-avian dinosaurs, and later birds possibly having this sort of negative correlation and potential competitive exclusion. And more generally, I want to make the model more complex and see how that affects the results. So the last section then is paleobiodiversity. How are we doing for time?
20 minutes.
Okay, it should be fine then. Okay. So I like to do, whenever I talk about biodiversity, I like to start by finding like the newest species named. Unfortunately, I did this a week ago for a different version of this seminar, but I'm just keeping it in anyway because it's kind of amusing. So there's this crab that was named for Harry Potter, so it's Harryplax severus. But the point is basically we still don't know how many species there are. We're still finding new ones. Modern biodiversity is vast. But at the same time, probably the most astonishing conclusion of Darwin's whole sort of origin of species is that ultimately all that diversity collapses down to a single common ancestor. So generally, macroevolution is kind of how did we get between these two extreme endpoints? And obviously, as paleontologists, we've got the only data set that tells us anything empirical. So really, in terms of paleobiodiversity, there's only two metrics that have sort of seen a lot of attention in paleobiology. The first one is simple taxon counting or richness, so things like the classic Sepkoski curve. We've also looked at morphological diversity, or as it's often termed disparity. This is a really nice sort of stack of ordination spaces from Mike Foote's paper. But I would argue that maybe we should look at other metrics, and I'm going to use a contrived example to show you why we might want to do that. So let's say we have these two samples here. As sort of biologists in a broader sense, I would hope that I could argue very convincingly to you that we want to consider the right-hand sample to be a lot more diverse biologically than the left-hand sample, because we've got a broader range of sort of types of organisms in there, modes of life, etc. But if we use something like richness, we would say, well, there's only 5 different kinds, they're basically equal. And if we wanted to do something like geometric morphometrics, we could set up a landmark system for these birds, but then we'd have big trouble applying those to things like a crab or a jellyfish. So one potential solution for this is to actually use a phylogeny, because everything can be placed in a phylogeny, and have that be timescaled. And so the answer would look something roughly like this. And then if we want to use a metric called phylogenetic diversity, all we're doing then is adding up those branches to give us a value in millions of years, and that would tell us what I think we want to say about these, and that's that the diversity of this sample is much larger than the diversity of this sample. So biologists use this kind of metric all the time. Here's an example of living mammals. So this is how phylogenetic diversity varies over space. The time scale here is billions of years. So basically, you're sort of giving a value in terms of sort of total evolutionary history in your sample. But if it varies over space, it probably varies over time, and that's something that might be interesting. So why have paleontologists not looked at that before? And probably the limiting factor is we don't have the ginormous trees that you need to do it. But as I've just argued in the first section of the course, now maybe we are. able to generate those trees. So again, I'm going to use the Dinosaur one, and actually I'm going to use 100 of the optimal trees. So I said right at the beginning, the Metatree and the formal supertree allow you to look at phylogenetic uncertainty, which is quite important in a more sensible way. And again, all the temporal data is coming from the Palaeobiology database. So before I show you the actual empirical data, what we did first of all, or what Dave did mainly, was generate a bunch of birth-death models to give us some kind of expectations against which we can compare our empirical data. So the simplest one is basically no births, no deaths. So we just end up with like a linear rise in phylogenetic diversity, and the slope is going to be dictated by the number of tips you stick in your starting tree. So that's probably not very realistic, but it's kind of a null that we might want to sort of compare things to. So then we looked at a bunch of constant rate birth-death models. So the top one would be a pure birth model, no extinction. The middle one, we've got birth being higher than extinction. Then we've got one where they're equal, but the asterisk kind of indicates that we're only keeping the simulations where the clades survive to the end. If birth and death are equal, sometimes obviously the clade can go extinct. But we find that it doesn't matter which one of these we use, we tend to see with every one of them that phylogenetic diversity sort of rises exponentially over time. Then we did a more complex diversity-dependent model. So our birth rate and our death rate were both dependent on our carrying capacity. We set it at 50 lineages. And then what happens is you get initially something like the exponential curve for the constant rate birth-death models. But then once the diversity dependence effect kicks in, you get an inflection point, and it starts to bend the other way, but it's never really asymptoting. So generally speaking, phylogenetic diversity is not going to reach an equilibrium, and that's just because of the way it's measured. So those are ways then we can sort of think about what data might look like under these different processes. And here's what we actually get when we look at dinosaurs as a whole. So we're going again from the Triassic on the left through to the end Cretaceous on the right. And we see initially it sort of follows that exponential rise, so more like the constant rate birth-death models. But then we see an inflection point, maybe more like the diversity-dependent model. But then we actually reach a peak and decline. So none of the birth-death models had this. Everything was always monotonic. And if we break down our tree into the sort of three major subclades, with ornithischians, we see a similar pattern, maybe where the peak is shifts further to the right. Sauropodemos, that's where all our phylogenetic uncertainty is, which is why the big error bars are there, and the peak shifts more to the left. And with theropods, we get more of a plateau, but basically, we're still getting a sort of prolonged decline in there. So this seems to agree with a recent paper by Manabu Sakamoto and colleagues, suggesting that dinosaurs had this prolonged decline prior to their extinction at the end of the Cretaceous. But I wanted to look at this in more detail. So the first thing we did is looked at richness, and this is expressed by number of lineages, or if you like, lineages through time. So this allows us to make direct comparisons between our phylogenetic diversity and our sort of species richness. And what we see is a very similar pattern. We see a rise to a peak somewhere in the Cretaceous and a prolonged decline. And if we actually do a correlation across our 100 trees, we see they're sort of all crammed up at the sort of the strong positive correlation end of a Pearson correlation coefficient. And we can do the same thing again with each of our subclades. And we get strong correlations. It's a bit weaker for Sauropodemos, but it's still fairly certainly a positive correlation. And this is potentially an issue for two reasons. So first of all, it might suggest there's no point doing phylogenetic diversity because actually it's telling us the same thing that richness is telling us. But the other thing is if it's correlated with richness, maybe it's biased by sampling. I think we're generally happy that richness probably shouldn't be considered without sort of correcting for sampling in some form. But I think we can kind of get around both of these things. So the first thing we can do is plot for just one of our trees our phylogenetic diversity on the y-axis against our number of lineages. So this is all of the sort of 1 million year time slices that we're looking at. And we can see that this is clearly not the kind of random redistributed residuals we might expect around a sort of linear line through the middle. So effectively, there's a systematic bias between richness underestimating and then overestimating phylogenetic diversity through the course of the Mesozoic. And this is true again of the subclades. There's clearly something else going on there. So this tells us that phylogenetic diversity is capturing some signal that richness is not capturing, so it is something that we might still want to look at. In terms of the second issue, a really simple way of dealing with this is to take richness out of it completely by subsampling our data. So what we actually did is subsampled it down to 10 lineages, so we're basically keeping richness flat right through the Mesozoic, and then we're only looking at variation in phylogenetic diversity. So if we do that with dinosaurs, what we get now is something much more like a linear rise. So this looks very like our null model, where we've got no speciation, no extinction. Obviously, we don't really think that's what's happening. Instead, though, what might be happening is something like what we get with rarefaction of richness, where we just end up with a flat diversity curve, which is only telling us really that we don't have enough data to resolve it. So that might be what's going on here, but actually this time, if we break down the data into our subclades, we see there are some interesting variations, so maybe we are able to capture something interesting. In particular, there's two declines here, one in Ornithischians and one in late Cretaceous one in Ornithischians, and a mid-Jurassic one in sauropodomorphs. So maybe subsampling is capturing some more interesting biological pattern that we can't explain by sampling or linked to richness alone. So the other thing, though, then is to go back and say, well, why do we see this decline? Is it real or is it an edge effect? So it doesn't show up in any of our birth-death models. So what's going on there? And the way I decided to try and deal with this is to add in a fake extinction, basically the point where our data looks more like a constant rate birth-death model, which what we're going to use here is basically a fake end Jurassic extinction. So there's two potential scenarios then. One is if it's a real signal, we ought to see the same kind of signal we see without the extinction in terms of the first part, so it ought to look more like this exponential rise. Whereas if it is an edge effect, what we might actually see is the same pattern just squashed further back in time. So we have a rise, an inflection point, a peak, and then a decline. And so if we do that with dinosaurs, we get rid of all of our Cretaceous data, pretend that's an extinction for dinosaurs, this is what we get. So the dashed line is basically the extinction. So we see, again, the exponential rise, an inflection point, a peak, and a prolonged decline. So a mid-Jurassic peak this time. So this very strongly seems to suggest that it is an edge effect. And we can do the same thing with our subclades again, and we still see basically some kind of decline in there. So that's kind of depressing then. Maybe we can't tell if we have this edge effect whether or not there really is a decline, because we're going to see a decline whatever. But if we go and look at the subsample data, maybe there is some hope. So if we subsample the data without the Cretaceous in, we get something more like the linear rise that we saw in the entire data set. We don't see a decline artifacts seemingly in here. And if we compare this to the data that does include the Cretaceous, so just truncating the same time series as before, the y-axis is extended here, but you look at the two time series, they look very similar. So this suggests subsampling may be a way of getting around this edge effect. And again, we can do the same thing with the different subclades. And in particular, we're still able to capture this mid-Jurassic decline in sauropodomorphs, and they rise up afterwards. So doesn't seem to be an edge effect in there. So to conclude sort of this section and the whole talk itself, effectively our birth-death models suggest that we should always see like a monotonic rise in phylogenetic diversity. That's not what we see with empirical data. We seem to have a decline in there. I would argue this seems to be an edge effect, so maybe this is something like the Signor-Lipps effect, but a special version for phylogenetics. But subsampling might be a way to get around this, and if we do subsample, we still see interesting patterns that we might want to explore in terms of what may explain them biologically. In particular, this late Cretaceous decline in Ornithischians and then mid-Jurassic decline in sauropodomorphs. And that's where I'll finished. Thank you.